Okay, so this first bit of the digestion, we're going to talk about kind of the actual act of digestion. Okay, so the basic idea here is we are taking polymers and breaking them down into monomers. Okay, so catabolism in this case. Now we talked about this a little bit with the stomach and how the stomach starts to break down proteins. Um, Most of the digestion takes place in the small intestine. Okay, so that's where most of the molecules are broken down even further. Okay, um, so first thing is there's enzymes, and the pancreas, which is a exocrine gland, um, releases enzymes into the small intestine as pancreatic juice. The pancreatic juice has a mixture of various things that help with digestion. Now, one of the things they have are these three enzymes: amylase, lipase, and proteases. So, amylase, um, more particularly in this case, pancreatic amylase, breaks down starch into smaller carbohydrates. Okay. Lipase, which breaks down fats into fatty acids and glycerol, and proteases, which break down proteins, which have already started to be broken down in the stomach into even smaller dipeptides, polypeptides, and amino acids. Okay, because it's only the monomers that get absorbed into the blood. Okay, so this is the whole point: is to get these things into the blood here so that we can use them for anabolic activities. So for building proteins, building muscle, building hair, um, the carbohydrates for energy, that sort of thing. Okay. Now the liver too. We're going to talk about the liver in a lot more detail later, but it also has a couple of functions with digestion. One is it secretes something called bile. Okay, and more specifically bile salts, which basically take kind of big fat pieces and break them down into smaller globules. Okay? And this is another example of a surfactant. So it's amphiphilic. It's got hydrophobic areas and hydrophilic areas. And you can see, if I zoomed in on this, the heads, the tails would be pointing towards the fat, while the tails would be pointing away, right? And that allows them to be broken down into smaller pieces, right? And also, and you kind of see that here, is what this does is it increases surface area, right? And when you increase surface area, you increase the rate of reaction, okay? And that allows lipases to more effectively break down lipids. The liver's got a whole bunch of other functions, which we'll talk about next year, but these are the functions particularly related to digestion. This works. If you have any questions, let me know. Thank you. Right, finally, just a little bit more about breaking down molecules, a little bit more visual, right? So here's a fat molecule. Remember, these are fatty acids. Can't escape this stuff. These are glycerol. And what lipase does is it breaks down the glycerol and the fatty acid. Okay? Um, this is an example of what we call an endopeptase. So it only breaks down in the middle, like exopeptases, for example, will, if this is a primary amino acid, endopeptases will break the ends only. Okay? And again, we just end up with two smaller peptides. These are broken down eventually into individual ones, into individual amino acids. These are what's absorbed. Okay. Then carbohydrates, we'll talk more in a lot more detail about these, but one thing is starch gets break down into maltose, which is 2-glucose, and those two get broken down into glucose molecules. Right? This is by another enzyme called maltase. Okay, so in a lot of cases, this is kind of a multi-step process of digestion. All right. So I hope that kind of explains the basics of digestion and what you're